Okay, any questions before we get started? So we're going to finish up with a reaction or two on alkylation, and then we're going to move into the aldol reaction and learn a little bit about bond polarity and counting functional groups by examining a natural product called discodermalide. So one other alpha functionalization called the Hell-Vollard-Zielinski reaction, so HVZ, uses phosphorus tribromide and bromine and water. So that's quite the witch's brew there. Each of PBr3 and bromine are quite nasty to handle. Essentially what happens is we substitute an alpha hydrogen for a bromine. And so now we have this alpha bromo acid. So it's a good leaving group. We can then do SN2 chemistry on it. And historically, it's been used to make alpha amino acids. So I'll put a couple arrows there. And so those are the functional groups used to make peptides. We'll get into that in Module 5. A couple different ways of making alpha amino acids. This is historically one way. It's probably not used too much anymore because alpha amino acids are enantiopure when they're in proteins, and so people want to make enantiopure things. This method gives you the racemic stereocenter at the alpha carbon. So what can I say about the mechanism? These are acidic conditions, so it's going to proceed through an enol. And then the Br2 is the electrophile that reacts with the enol. So not much more I want to say about that. So in terms of alkylation, where we're treating with an oxidation state 1 electrophile, and then the beta carbon ends up at oxidation state 0. Pay attention to the base that's used in the reaction. So with this unsymmetric ketone, you have alpha and alpha prime, and then sodium hydride again is going to form the thermodynamic enolate. So that's the one that's more substituted. And then we're treating with benzyl bromide, so that's a good electrophile. And that H that's there gets substituted for this whole group. So maybe one quick question. This this carbon right there that we did the substitution, is that a stereocenter? So I see some no's. It's not, because the ring is basically symmetric. So that's not a stereogenic center. Just I'll have a couple of these brief detours throughout today's lecture. The molecule shown on the left is called S-adenosylmethionine. You may have heard it on like supplement commercials. It's called SAMe. So people take it for things like depression and anxiety and stuff like that. And so SAMe is what nature uses to put a methyl group on something. So this is an electrophile, and this methyl group is electrophilic. So what you can think of that sort of biological molecule as. It's the equivalent of iodomethane. So in lab, we would use iodomethane as an electrophile for one carbon, and then nature uses this SAMe. So everything bonded to the methyl group is just your leaving group. And then presumably that can get recycled back into SAMe and react again. So the fundamental reaction of the substitution is sort of the same mechanism, but under physiological conditions, you have these other reagents that are more compatible with biological systems. So as we'll see with the aldol reaction coming up, nature's making sort of the same bond connection, but the reagents employed are different. And then we'll end this on this term vinylogous acidity. So remember that we said we were only focusing on the alpha carbons. However, if you have a double bond next to a carbonyl, so a carbonyl and then an alkene or unsaturation, you can have something called vinylogous. And so if you look at this molecule shown, this cyclopentenone, if you go around the ring counterclockwise, as I'm showing in red, you see that alpha prime and beta prime are both sp2 hybridized. So these H bonds, they're not very acidic. If we go around the other way, clockwise, there's your normal alpha carbon, so those are acidic. And then when we meet that fifth carbon, depending on which way you go, either beta or gamma prime, those hydrogens, even though they're beta, the one way, they're gamma the other way, those are actually pretty acidic as well. And so we can think of a reason why that is. So let's ask a question here. So if we treat that molecule with NaOD, sodium deuteroxide, and D2O, how many deuterium atoms are taken up in the molecule? So we're starting with C5H6O. So we know that at least two of them will incorporate deuterium. But then what about these other positions? So maybe I'll draw an intermediate. I'm just going to swap out the two alpha H's. So we know we can deprotonate those and then exchange them for deuterium with the D2O. And then ultimately, I'll give you a hint. We end up with a molecular formula of C5H-D5O. So if I call that alpha, those beta hydrogens are acidic because of that double bond. And so what will happen if we show the sodium OD, abstracting one and putting the negative charge on that carbon. That's stabilized through resonance from that double bond. So the negative charge exists there, and then through resonance, we can push it onto that alpha carbon. And so that resembles the normal, one of the forms of an enolate. So if we just push the arrows again, that would draw the enolate form. And then what you can imagine is that, so this negative charge here, if that just reacts with D2O, it's swapped out one. You have this other H, the same thing can happen. So that'll swap out for two more. And then in this resonance form, you have that negative charge there. So that H can get swapped out for D. And then ultimately, the only unlabeled H that remains is on that other sp2 hybridized carbon. Yep.
Oh, I didn't get in the last step there were two double bonds, and then in the product there are only one double bond. Can you explain that again, please?
So when this comes down, it's going to go, so that's reforming the ketone. This comes back, and then that can get deuterated with D2O. So this molecule, I think, has some deeper meaning to us, and I'll just sort of explain two things about it. So here's alpha. So we'll say alpha prime, beta prime. So it's an unsaturated ketone, and these are called enones, E-N-O-N-E. So we'll learn how to make those using the aldol dehydration reaction. And then once we've made those, we're going to finish this molecule off with another type of reaction called a Michael addition. So the counting of locations of functional groups along the carbon backbone, we'll start to see a pattern to them. So the take-home message here, if you have that enone, that gamma carbon is acidic. So that's vinylogous acidity due to that possibility of resonance. Any questions? So we have 60 minutes and 45 slides to get through. We're probably not going to get nearly halfway through that because the aldol is pretty complex. There's a lot of things we have to consider. It's one of the most studied reactions due to the complexity. And I want to put it into a little bit of context so that you can see the logic that's at play. So I'm going to show you a natural product. It's called a marine natural product because it comes from a sponge. So you have these scientists that throw on some scuba gear. They go underwater exploring to identify these organisms. They harvest them. And then basically they test extracts. So you have this sponge. You lyophilize it. Does anyone know what that means? Basically, you freeze it, and then you put it under a vacuum, and it sucks the water out. So it's called freeze drying. You probably all drink Starbucks, so you're not going to get this, but you go to Walmart in the coffee aisle, there's like this brick of coffee. It's usually like freeze dried, and so that's what they're doing. They're taking all the water out of it. And so once they get the dry sponge, they just extract it with organic solvent. So they get these different extracts and they test it against, like, say, cancer lines. And then if one of those extracts is active, then they can go back and figure out what's causing the activity. So the molecule we'll see is called discodermalide, and it's a cytotoxic or anti-cancer agent. And so these natural products, they come from plants or living organisms, and they're pretty complex. And you can ask the question, why is a plant producing such a thing? What's the need? There's a video on YouTube by Professor Paul Jensen. It's called From Sea to Pharmacy. It's about 50 minutes long. But since most of you are biology or biomedical sciences majors, I think you'll find it pretty interesting. There's a lot of different areas of science that he talks about and how these living organisms produce these molecules that can become drugs. And so like the expedition of doing that. And there's a lot of really neat experiments about, why the organism's producing this. I mean, I don't know much about like trees, but like trees don't get cancer, do they? So why is a tree like producing such a complex molecule? It's like one reason is for defense. Like if you don't want insects eating it, it excretes this compound and then the insects like don't eat it. So I think he's at Scripps. Have you heard of Scripps? These oceanographic institutes, they're pretty famous. Cold Spring Harbor is another one. Woods Hole and then Harbor Branch are four very well-known scientific research organizations. So they study a lot of this fundamental science, and so part of that is these natural products. So the one we'll look at is called discodermalide, and it's an immunosuppressive and antifungal agent. People call it disco in the literature, and there's Disco Stew from The Simpsons. It was isolated in 1990 from this organism here, which I'm not going to even try to pronounce, by Gunasekara. And it's pretty complex, right, when you first look at it. But if you take small bites out of it, it's not really that bad. You can identify the functional groups. You can start talking about stereochemistry. And then what we're going to focus on is the relationship of the functional groups to one another. So again, that molecule is made in that organism. So the organism does not have round-bottom flasks. It doesn't call up Aldrich and order chemicals. It's coming from, these biological molecules. Attracted a lot of attention in the literature, And so again, this is sort of an aside about biochemistry. So if you go on to, say, biochem or cellular and molecular biology, you'll study these metabolic pathways. And so this is just the screen grab I got called the metabolic metro map. And really what it is, you have a small starting material and you're tracing where carbons go, how things get oxidized, how they get reduced. But you see it's pretty complex. And so, like, for example, like carbon fixation. So, like, what does that mean? So, like, plants take up CO2, right? And then they convert it into an energy source. So that's carbon uptake, carbon fixation. There's things like nitrogen fixation with fertilizers and stuff like that. So, it looks complex, but it gets even more complex because in each one of these like loops, there's a series of reactions that happen. So one of the two molecules that nature uses to install two carbons is called acetyl coenzyme A, or acetyl-CoA. And as I said, nature doesn't use like something we would use. So for example, ethyl acetate. So there's an ester. If we focus on these two carbons right here, so that's derived from ethanoic acid. You see all this is ethanol. So what nature uses is like those two carbons, but all this whole other thing is just equivalent to that ethanol. And what we're studying is the chemistry of the alpha carbon. So again, conceptually, what you're learning now, nature does sort of the same thing, but with these larger molecules. So acetyl coenzyme A has those three sort of units to it. And really what it is supplying two carbons to a molecule. For three carbons, it's propionyl coenzyme A. So propane is 3, so that's the 3 carbon unit that you can see right here at the end. The other thing that's different is, see this bond right here? That's a thioester. It's not a normal ester. So it turns out that's a better leaving group under physiological pH. And the reason for that is if we look at sort of two small organic molecules comparing just the normal ester oxygen versus the thioester with S, cleaving that bond is much easier by about 100 kilojoules per mole. And so as we ultimately get into Module 5, we'll see how nature uses these other heteroatoms. So sulfur used quite a bit in place of oxygen. Phosphorus is used quite a bit in place of nitrogen. Maybe one reason for that is, again, they're below. So they're in, group 3? So if we have CNOF, silicon, phosphorus, sulfur, chlorine, that's period 2, that's period 3. And so those have extra orbitals available to them to do more complex things. So these natural products are often called secondary metabolites. So they're produced by the species, but they're not normally used for growth, development, or reproduction. So they're used for things like defense, or if I'm getting this concept right, quorum sensing. So if there's like one little dude here and he wants to attract his friends, he spreads out this chemical so they all come around him. So there's like defensive purposes and attractive purposes. So secondary metabolites can be produced through enzymes. So that's the biological reactants, enzymes. And one route is called polyketide synthetases. And so these PKSs have these modules, which are collections of domains, and then the domains carry out individual reactions. So on this link, which I have a screenshot on the next slide, is going to show you how complex this can actually get. The modules start or load, elongate or extend, and then terminate or release. So any molecule produced in nature it's for the most part going to come from those 2 or 3 carbon units in a long chain. And then if it cyclizes, it can do that, but then it's ultimately released from this enzyme. So this is a screen grab from Roche, so that's a pharmaceutical company. And it's much clearer on the screen than it is on my iPad. So here's the acetyl coenzyme A right there. And basically when you follow these things, these metabolic pathways, you're tracing where carbon atoms are included or incorporated. And then there are steps that do oxidations, reductions, or even excise them. So like for example, you've heard of glucose, like the main sort of sugar that we eat. So glucose is 6 carbons, and it's a form of energy. It gets metabolized, and then if everyone take a deep breath in, and then exhale. So what are you exhaling? Carbon dioxide. So where does the carbon come from? It comes from glucose. So those 6 carbon atoms of glucose eventually get oxidized and then chopped off the chain, and you exhale them. So fun fact, if you have a bathroom scale, weigh yourself tonight and then weigh yourself when you wake up tomorrow morning. You'll probably be 1 to 3 pounds lighter tomorrow morning, just by cellular respiration, all that CO2 you're breathing out. So athletes typically lose more weight overnight because they're more active. So a professional athlete can lose like 5 pounds overnight. It's pretty wild to think about. So even though this looks complex, it's really just all the reactions we've studied so far. So let's take a look at discodermalide and reduce its complexity through some simple operations and observations. The first thing I want to do is see this ring here. It's called a lactone. That's a cyclic ester. If we cleave this bond, that's going to get us a carboxylic acid and an alcohol. So that operation produces what's called a ceco acid. It's just the straight chain form of a lactone. So we've got that out of the way. And so now let's look at the region in red that's boxed. There's 6 carbon atoms there. Where do you think those 6 carbon atoms come from? The acetyl coenzyme A or the propionyl? So remember, this supplied 2 carbons, this supplied 3. So I'll highlight in red. So see these 3 carbons, and then these 3. So those come from propionyl coenzyme A. If we use the carbonyl carbon as our anchor point of 1, and then we walk over to that red arrow, what number should be there? So 1, 2, 1, 2, 3. So the relationship from the acid to that alcohol is 1, 3. And we've already seen that the alpha carbon is next to the carbonyl. And so that's the beta carbon. So this is a beta hydroxy acid. If I restart numbering, so I'm at the beta carbon now. If I start that as 1, and then I count over 1, 2, 3, there's another alcohol. Then I count over 1, 2, 3. Then I count over 1, 2, 3. Whoop, what happened? So if you look, if that was an alpha carbon and that's a beta carbon, if this was an alcohol and that was an H, that's a dehydration. So wherever you see those alcohols, they can basically eliminate water to form these double bonds. And then you can just do the same thing, 1, 2, 3. And then there's some weirdness here. 1, 2, start of a double bond. So within that polyketide synthetase, there's a reaction that happens that's sort of unnatural, because you would expect the double bond to be right here. So there's this unnatural bond disconnection. So just to put some of this in historical context, there's logical ways of describing this. So at Harvard University, there's a professor, E.J. Corey. He published a book, The Logic of Chemical Synthesis, where you're examining these functional group relationships. The professor shown here is David Evans. And his way of thinking about it was called charge affinity analysis. And I like thinking about it in that terms because it's just electronegativity and how you make bonds based on that. So this is a chemist tree, if you will. So here's David Evans. And my PhD advisor, David Hart, did a postdoc under David Evans at Caltech. Evans eventually moved to Harvard. So I studied under David Hart at Ohio State. So I'm right here. Now I have my own group, my own small group. If you look at all these are all Hart students. I think I was number 40. His 40th PhD in the list there. I think he ended up with like 44 or something. This other guy at Ohio State had over 100 PhD students, so that's a very productive career. If you're unaware, all of you are undergraduates, and so if you decide, you can go to graduate school, so that's when you do research. Then if you're in for more pain, you can go on to do a postdoc after you do your graduate studies. But some of you will go to like professional schools, that's like med school, dental school, that stuff. That's not research based, so. But any event, I didn't get my first real job until I was 31. So you're young, hang in there. Your 20s are not the end of your life. So the connection between this is, if I have it up here, yeah, right here. So Evans earned his PhD with Ireland. And so all of these guys were interested in this aldol reaction, and they're really the ones that took it from sort of this rudimentary thing to this really rational place where you'll see as it increases complexity, they figured out how to control all the elements involved. So the charge affinity analysis, if we just look at propanoic acid at the top here as our model, and we know that from an acid, the H is acidic. So I'm going to just assign it that plus charge. And when we deprotonate it, we know that the oxygen can be negative. We know from a carbonyl we can do a nucleophilic addition, so that's positive. The pi bond breaks, and so the oxygen, or the carbonyl, is negative. We know that the alpha carbon hydrogens can get deprotonated, so that's negative. And then what you see is just the charges alternate along the backbone. And then I filled that in for the CHE256 acid of discodermalide. So in that, again, here's your alpha carbon, and then there's your beta carbon. So the beta carbon, as it's shown right here, is in oxidation state 1. Now, if we think about how we can make that, it has to come from a higher oxidation state. So that would be 2. The other thing bonded to it is a hydrogen, right? So if it's at oxidation state 2, that has to come from an aldehyde. And then the alpha carbon is coming from an enolate. And so when we make that bond disconnection between alpha and beta, where beta is an alcohol, that's called the aldol reaction. And so ultimately, this molecule has been made just using variants of the aldol reaction. So it was a pretty hot molecule back when I was an undergraduate. Actually, no, I was in high school in '93 when it started. So from '93 to 2004, you can see all these different groups worked on making this molecule. And the reason is, even though it's complex, it's so potent that you don't really need much to treat cancers, like nanogram type amounts. And so these groups worked on strategies to make them, you can see the longest linear sequence. What's like 29 is the highest here. So 29 sequential steps. Total steps, 49. And then you can see the overall yields are pretty low, but still, if you get ultimately, say, a gram of this, divide a gram by like 5 nanograms, that's quite a lot of doses, right? So you can do a lot of clinical studies with it. So we're going to look at the basics of the aldol. We're going to start with the 2-carbon aldehyde, so ethanol. We'll think about it under basic conditions, so where we generate an enolate. This can also happen under acidic conditions with the enol. But we're going to see the complexities of this. So from ethanol, then we're going to go up in complexity pretty quick. So we'll just write some stuff out, and then there'll be some questions about it after. So I want to do this. This is called a self-aldol. So we just have the 1 carbonyl component in there. So 2 molecules of that are going to react to form that product. So under basic conditions, I'm just going to show one of those hydrogens of the methyl group. So let's just say sodium hydroxide in water. So we're going to form our enolate. That's going to be the nucleophile. I'm just going to keep labeling that alpha so we can track things. So enolate, so that's the nucleophile. Now it's going to react with just the aldehyde itself. And so I'm going to put under there beta, because that's going to become the new beta carbon. And the enolate will collapse to reform the carbonyl. The alpha carbon attacks, that's the nucleophilic addition. And then we break that pi bond. So we're making a bond between alpha and beta. And then the total chain length is 4 carbons. And then we can just do a proton transfer with water. And so this is a beta hydroxyaldehyde. Again, the key thing for this module is we're really focused on the alpha and beta carbons because that's the bond we're making. And then if we look at the beta carbon, we see it's oxidation state 1. So that is telling us it was made from the aldol reaction. Yep.
Is it called the second or the beta carbon because it's second from the carbonyl?
Yeah.
Okay.
So I think these are just, so what was the nucleophile, what was the electrophile? So remember, we're dealing with 2 carbons only. So A has to be wrong because that has 3. Then we want an enolate for the nucleophile. So C has to be wrong because that's an enol. D has to be wrong because that's showing the electrophile. So our answer for this would be B. So in the aldol, we have to identify what's the nucleophile, the enolate part, and then what's the electrophile. And what I'm saying holds true for ketones as well, even though I'm not going to lecture you on those. Replace the H of the aldehyde with a carbon, you can do the same chemistry. Couple questions. Yeah. So 3-hydroxybutanal. I want to say something about the naming again. So it's 4 carbons that comes from butane, and then we have 2 functional groups, the aldehyde and the alcohol. The aldehyde is at oxidation state 2, the alcohol is at 1. Since 2 is the higher oxidation state, that gets the parent name, so that's why it's called butanal. And then the alcohol is the substituent. So 3-hydroxybutanal. It says, does the molecule contain any stereogenic carbons?
Yes.
So yes. So with the asterisks, the beta carbon is a stereocenter. So the next question is, those are the 2 enantiomers. What ratio would they be formed in? So they have to be formed in a 1-to-1 ratio or 50-50, because you're starting from stuff that is achiral, and then your product mixture has to end up as achiral. So we get C for that. But this is highlighting to you that immediately the complexity already. Just even from a simple 2-carbon aldehyde, you're generating something with a stereocenter, it's a mixture. And if you start thinking about, well, I only want the R enantiomer, how do I do that? It gets pretty complex quick. But there's ways of controlling it. So as I said, when you see these double bonds, a double bond you can think of as coming from the elimination of water across the alpha and beta carbons. So there's your beta hydroxy aldehyde. The alpha carbon has an H, the beta carbon has an OH. And so we can eliminate that to get an alkene. And so this is called an enal. So an alkene aldehyde, enal. And it says that geometry is E, which is equivalent to trans. And so why does it favor the trans? So let me put my thinking cap on, see if I can remember the way to draw this. The thing we're going to use is a Newman projection. So remember, Newman projections show us dihedral angles. That H and OH have to be 180 degrees away from one another, or anti. And so, again, what's tricky about this, I said these alpha H's were different, right? So you'll see that they're different when we align them relative to the OH, because one will eliminate to trans and the other will eliminate to cis. So I think I'll stick my eyeball right here. So that's my eye, and I want to look down alpha to beta. So alpha's in front. I'm going to draw the aldehyde carbon up, and then our two hydrogens. So our back carbon is the circle, and then it has say OH, H, and then carbon 4 is the methyl group. So I think I kind of got lucky here. There's the OH of the beta carbon. And then you have H and H. This H right here is the one that gets eliminated. Trying to think of a way I can draw this in a Newman projection that's not going to look weird. So the CHO is still up. And then, now you have to imagine we're looking down a double bond now instead of a single bond. So that H is going to be down. And then the back carbon, the H is up and the CH3 is down. So if we unfold that, it should look like that. So I said the two H's were different. If we rotated this other H to put it anti to the OH, we could see what happens. So I'll sort of box this area off. I'm going to do a rotation now. So I'll highlight that one in yellow. So yellow and then blue, that was our rotation. So the issue now is for that H to get deprotonated, see this interaction here? That's a gauche interaction, so that's destabilizing. And that would lead to the cis alkene, which is less stable than the trans. So the aldol product itself, the beta hydroxycarbonyl, that's stable. But under forcing conditions, you can dehydrate it. And then the trans geometry of the alkene would be favored. Any questions? We have 20 juicy minutes left. This is where it's going to get more complex now. So we're going to do a cross aldol. And so that contains two different aldehydes. And what we have to suss out is all the products that are formed. So acetaldehyde or ethanal plus the 3-carbonaldehyde propanal. Now just looking at it, you're treating it with sodium hydroxide and water. What could be a question you would ask first?
Which one's deprotonated?
Which one's deprotonated? So which one forms the enolate? So they're both going to form the enolate. And then the follow-up question is, which one's the electrophile? They're both going to act as electrophiles. So you're doing a cross aldol in two senses. This can be an enolate reacting with that. That can be an enolate reacting with that. And then these things can still do their self-condensations with one another. So you basically have 4 different pathways that can be followed under this, under these conditions. If you did not read ahead and you had to take a guess, how many total products do you think are going to form? 3? Any other brave souls? So 3 is quite low. We have an 8. So I'll just tell you what it is. You can form 12 products from this. So remember that the self-aldol with just ethanal formed 2, right? Because of your R and S. Now you have these 4 different possibilities that are all producing their own unique products with stereoisomers. So you can get like a mess. Like if you want to make one thing and you get 12 possibilities, how do you control all that? So we'll eventually see how we do that, but let's just figure out what these products are. And this is all written on the next slide or two, but we'll walk through the logic. We'll say electrophile, and then enolate. And then we have 4 scenarios. I'll say cross, self, and self. So in the first one, we'll just write it sort of as the reaction's written, where the ethanal is the electrophile, and then the enolate is coming from the propanal. So remember alpha, beta. We're just going to form that alpha-beta bond. So now we have 1, 2, 3, 4, 5 total carbons in the chain. Excuse me, 4, because it's branched. I feel like a horse's patoot. So it's 4 because there's branching from the enolate. So there's alpha, there's beta. So looking at that product, how many stereocenters are there? there's 2 stereocenters. So how many total stereoisomers? So right now we've shot past 3. So for the other one, we're just going to flip the electrophile and enolate. So now the 3-carbon aldehyde is the electrophile. The 2-carbon is the enolate, and so we can draw that product where we keep track of alpha and beta. And we see that that only has 1 stereocenter. So the asterisk is a stereocenter. So that gets us 2. So we're up to 6. Now we can do the self condensation with the ethanol. So we've already seen that in the first example. So that has 1 stereocenter, so that's 2. And then the last pathway is the self with the propanol. And again, that has 2 stereocenters. So total, that's going to give us 12 possible products. So, I mean, you can go through the normal unique carbons, unique protons, their sets, and then it gets more complex because, you know, with your stereoisomers that have 4, and you think about, you know, those relationships, how do you actually figure out, you know, if a stereocenter is R versus S? You can look at Newman projections, dihedral angles, and coupling constants can help you figure that out to some extent. Any questions before we move on? So this was, the total number of pathways was D. You can get 2 from the self and then 2 from the cross. So 4 possible pathways. Self-aldol products, what are their names? I think we did one of these already. So that was 3-hydroxybutanal. So I think I only see that in choice B. Then the other self product. So we have 1, 2, 3, 4, 5. So that's the pentanal, that gets the priority. Then we name the substituents according to alphabet. So 3-hydroxy and then 2-methyl. So that's a little bit of nomenclature review. Cross aldol, what are they? Let me think. So I think that was 1. So 1, 2, 3, 4. So that's going to be a butanal derivative. So 3-hydroxy-2-methyl-butanal. I think this ends up being A. Does it? Yeah, it ends up being A. So 1, 2, 3, 4, 5. Is that right? Oh, sorry. This should not be there. Sorry. So that's the second cross product. So that's the 3-hydroxypentanal. So one of the things you can do is look at a structure and determine if it comes from a self or a crossed aldol. And you'll be able to see that based on, the things bonded to the alpha carbon and then the rest of the chain. We said the total number of stereoisomers was 12. So we drew the products, figured out the stereo centers, used the 2N rule. Yep.
The question only refers to cross aldol.
Yeah. So I mean to change that every semester and I don't. So I'm like, change it. I'll go back to my office and promptly forget about it. But the intent is to mean 12. So let's just write, fix this. Will ya? Probably not. So here they are summarized. And let me just put the asterisk by the stereo centers. And so again, that's meant to show you that it's pretty complex, yeah.
Oh, which carbon from the two components right there forms that methyl group?
So that's how we can trace it. So again, what gives rise to all these products? There's a way of describing the face of a carbonyl using the CIP rules, but now the carbon itself is our anchor point. So if we look at the electrophile, which I have listed as carbonyl functional group, and so we're going to anchor our eyeball on that red dot. So that's an sp2 hybridized carbon. We can still apply the CIP rules using atomic number. Oxygen would get a priority of 1, carbon 2, hydrogen 3. So there you're going counterclockwise. And so as you're looking at it from like this trajectory, that's called the C face. If you look at sort of the opposite or mirror image of it, now you're going clockwise, and that's called the re face. And then you can do the same thing for the enolate where you're focused on that sp2 hybridized carbon. You can get the Re and C faces. So chemically, what is that telling you? One face of the enolate can attack one face of the carbonyl. And so since each has two different faces, you get the combinations of those, unless you can control that somehow. So let's do a little review here, see what we got. Really finish up on these questions before we get any further, but let's just look at this one to review those nomenclature rules. So we said there was two stereo centers marked by the asterisk. Yeah, we'll so 2 to the n, that's 2 to the 2, that gets us the 4. And then we'll consider those 4 for the next run of questions here. So in terms of the numbering, this is 1, 2, 3, 4, and 5. And the question is asking you, what is the stereochemical assignment at carbon 2 and 3? So we have, let's see. I like to do the thing with the attached heteroatom first because it's easiest. So I'm going to do carbon 3 first. So this would be, I'll use a different color here. So that's priority 1, 2, and 3. So that definitely has to be R. So 3s is out. Then in green, we'll do the alpha carbon. So that's 1, that's 2, that's 3. So that should be S. So yeah, it is B. So again, a subtle thing, when you're looking at that carbon and you go out, you have carbon and carbon. This one wins because you think of that double bond as 2 single bonds to oxygen. So oxygen and then oxygen, whereas this one only has 1. So that's why that gets priority. So 2s, 3r. Pay attention to the alpha stereochemistry. So the beta is staying the same, and the beta is staying the same, so that has to remain the same. So 3R, then we're flipping the alpha. The alpha is changing. So what should this be?
RR.
Do what?
RR.
Yes, you're correct. I was inverting them in my mind. Yeah, so this should be A. So if you look at them, alpha from S to R with that wedge to dash. So this is going to be A. I think we'll wrap it up here. Just a couple more of these check-in questions. And then next time I'll just give you a brief overview of how do you go from 12 to 1. It can be done. So people have figured that out.